Hello, students. Welcome back to BioDecode, where biology is decoded into simple steps. This is your trusted audio guide for learning biology the smart way. You're listening to the NCERT Class 11 Biology Audiobook, Chapter 4, Animal Kingdom. Before we dive in, if you haven't already listened to Chapter 3, Plant Kingdom and its exercise solution, you can find it streaming on our channel. It's a great way to build a strong foundation, as we'll be building on some structural and evolutionary ideas introduced there. In this chapter, we'll journey through the fascinating world of animals, exploring their diversity, body plans, levels of organization, and the key traits used for classification. You'll learn how zoologists group animals based on symmetry, germ layers, coelom formation, and modes of nutrition and reproduction. From simple, radially symmetrical forms to the highly organized bilaterians, we'll examine how each phylum represents a unique adaptive strategy in the animal kingdom's evolutionary history. This audiobook is designed to make each concept clear, logical, and easy to remember. Whether you're revising for your school exams or preparing for NEET, you can listen, learn, and retain the information effortlessly. So, plug in your earphones, focus your mind, and let's begin decoding the remarkable diversity of Animal Kingdom. This is BioDecode, and you're listening to Chapter 4, Animal Kingdom. Chapter 4, Animal Kingdom. When you look around, you will observe different animals with different structures and forms. As over 1 million species of animals have been described till now, the need for classification becomes all the more important. The classification also helps in assigning a systematic position to newly described species. Basis of classification. In spite of differences in structure and form of different animals, there are fundamental features common to various individuals in relation to the arrangement of cells, body symmetry, nature of coelom, patterns of digestive, circulatory, or reproductive systems. These features are used as the basis of animal classification, and some of them are discussed here. Levels of organization. Though all members of Animalia are multicellular, all of them do not exhibit the same pattern of organization of cells. For example, in sponges, the cells are arranged as loose cell aggregates, that is, they exhibit cellular level of organization. Some division of labor activities occur among the cells. In Coelenterata, the arrangement of cells is more complex. Here, the cells performing the same function are arranged into tissues, hence is called tissue level of organization. A still higher level of organization, that is organ level, is exhibited by members of Platyhelminthes and other higher phyla where tissues are grouped together to form organs, each specialized for a particular function. In animals like annelids, arthropods, mollusks, echinoderms, and chordates, organs have associated to form functional systems, each system concerned with a specific physiological function. This pattern is called organ system level of organization. Organ systems in different groups of animals exhibit various patterns of complexities. For example, the digestive system in Platyhelminthes has only a single opening to the outside of the body that serves as both mouth and anus, and is hence called incomplete. A complete digestive system has 2 openings, mouth and anus. Similarly, the circulatory system may be of 2 types. First, open type in which the blood is pumped out of the heart, and the cells and tissues are directly bathed in it, and second, closed type in which the blood is circulated through a series of vessels of varying diameters, arteries, veins, and capillaries. Symmetry. Animals can be categorized on the basis of their symmetry. Sponges are mostly asymmetrical. That is, any plane that passes through the center does not divide them into equal halves. When any plane passing through the central axis of the body divides the organism into 2 identical halves, it is called radial symmetry. Coelenterates, Ctenophores, and Echinoderms have this kind of body plan, figure 4.1a. Animals like annelids, arthropods, and so on, where the body can be divided into identical left and right halves in only one plane, exhibit bilateral symmetry. Figure 4.1b. Figure 4.1a. Radial symmetry. In this diagram, the organism's body is shown arranged around its central axis, like spokes radiating from the hub of a wheel. Free plane passing through the central axis divides the body into two identical halves. This type of symmetry is common among aquatic animals such as Hydra, Aurelia jellyfish, and starfish during certain life stages. Radial symmetry allows these organisms to interact with the environment from all sides equally, an adaptation suited for sessile non-moving or free-floating lifestyles. It ensures sensory structures and tentacles are evenly distributed around the body, which helps in food capture and defense, regardless of the direction of stimulus. Figure 4.1b, Bilateral Symmetry. Here, the figure demonstrates a body plan where only a single plane, called the median sagittal plane, can divide the organism into two identical halves, left and right. This symmetry is seen in higher animals such as annelids, arthropods, and vertebrates. Bilateral symmetry marks a major evolutionary advancement because it allows cephalization, or concentration of sense organs and nervous tissues at the anterior end, leading to directional movement and complex behavior. It signifies an active lifestyle where the animal moves in a definite direction and requires a coordinated body design. Diploblastic and triploblastic organization. Animals in which the cells are arranged in two embryonic layers, an external ectoderm and an internal endoderm, are called diploblastic animals. For example, Coelenterates. An undifferentiated layer, mesoglia, is present in between the ectoderm and the endoderm, figure 4.2a. Those animals in which the developing embryo has a third germinal layer, mesoderm, in between the ectoderm and endoderm are called triploblastic animals, Platyhelminthes, 2 chordates, figure 4.2b. Figure 4.2, germinal layers, a diploblastic, B, triploblastic. This figure compares the early embryonic organization of animals based on the number of primary germ layers formed during development. In the diploblastic structure A, the embryo has two layers, the ectoderm outer layer and endoderm inner layer, with a non-cellular jelly-like mesoglia in between. Examples include Hydra and Obelia. In contrast, the triploblastic structure B shows the presence of a third layer, the mesoderm, sandwiched between the ectoderm and endoderm. This additional layer leads to the formation of complex organs and organ systems. Animals like annelids, arthropods, mollusks, and chordates are triploblastic. The transition from diploblastic to triploblastic organization represents a key step in evolutionary complexity in body specialization. Coelom. Presence or absence of a cavity between the body wall and the gut wall is very important in classification. The body cavity, which is lined by mesoderm, is called coelom. Animals possessing coelom are called coelomates. For example, annelids, mollusks, arthropods, echinoderms, hemichordates, and chordates, figure 4.3a. In some animals, the body cavity is not lined by mesoderm. Instead, the mesoderm is present as scattered pouches in between the ectoderm and endoderm. Such a body cavity is called pseudocoelom, and the animals possessing them are called pseudocoelomates, for example, Aschelminthes, figure 4.3b. The animals in which the body cavity is absent are called acoelomates, for example, Platyhelminthes, figure 4.3c. Figure 4.3, diagrammatic sectional view of body cavities. This comparative diagram illustrates three types of internal body cavity arrangements. Coelomate, the true body cavity, coelom lies between the body wall and gut, lined by mesodermal tissue on both sides. It provides space for organ development, movement, and cushioning. Examples include annelids, mollusks, arthropods, and chordates. Pseudocoelomate. The body cavity is present but not completely lined by mesoderm. Instead, it lies between the mesoderm and endoderm. Found in roundworms ascaris, it allows limited organ movement and serves as a hydrostatic skeleton. The coelomate. No body cavity is present. Mesoderm fills the entire space between the body wall and foot. Flatworms planaria. Pania are typical examples. This figure highlights how the evolution of a coelom improved internal organization and organ efficiency. Segmentation. In some animals, the body is externally and internally divided into segments with a serial repetition of at least some organs. For example, in earthworm, the body shows this pattern called metameric segmentation, and the phenomenon is known as metamerism. Notochord. Notochord is a mesodermally derived rod-like structure formed on the dorsal side during embryonic development in some animals. Animals with notochord are called chordates, and those animals which do not form this structure are called non-chordates. For example, Porifera to echinoderms. Classification of animals. The broad classification of Animalia, based on common fundamental features as mentioned in the preceding sections, is given in Figure 4.4. The important characteristic features of the different phyla are described. Figure 4.4. Broad classification of kingdom Animalia, based on fundamental features. This figure presents a schematic tree showing how the vast diversity of animals can be systematically grouped based on key structural and functional traits. The classification starts from basic levels like cellular and tissue organization, body symmetry, and presence of a coelom, progressing to complex traits like segmentation and notochord development. The figure emphasizes the evolutionary transition from simple asymmetrical sponges Porifera to more complex bilaterally symmetrical coelomate chordates. Each branching point represents the emergence of a new structural innovation that defines a distinct phylum. This evolutionary roadmap helps students visualize how animal complexity evolved step by step over time. Phylum Porifera. Members of this phylum are commonly known as sponges. They are generally marine and mostly asymmetrical animals. Figure 4.5. These are primitive multicellular animals and have cellular level of organization. Sponges have a water transport or canal system. Water enters through minute pores ostia in the body wall into a central cavity, spongiocule, from where it goes out through the osculum. This pathway of water transport is helpful in food gathering, respiratory exchange, and removal of waste. Choanocytes or collar cells line the spongiocule and the canals. Digestion is intracellular. Body is supported by a skeleton made up of spicules or spongin fibers. Sexes are not separate hermaphrodite. That is, eggs and sperms are produced by the same individual. Sponges reproduce asexually by fragmentation and sexually by formation of gametes. Fertilization is internal, and development is indirect, having a larval stage which is morphologically distinct from the adult. Figure 4.5. Examples of Porifera. Sycon Euthpongia C. Spongilla. This figure displays the characteristic body forms of sponges, organisms belonging to the phylum Porifera, the simplest multicellular animal. Sycon sypha has a vase-shaped body with numerous pores called ostia on its outer wall, leading to internal canals. The canal system filters water and extracts food particles, demonstrating the sponge's unique filter-feeding mechanism. Euspongia bath sponge possesses a soft, fibrous skeleton made of spongin fibers, which allows flexibility. It represents the commercial sponges used for bathing. Spongilla, a freshwater sponge, unlike most marine species, forming greenish colonies due to symbiotic algae. This figure highlights the transition from simple cell aggregates to organisms with functional coordination via a canal system, an evolutionary step toward tissue-level organization. Examples: Sycon sypha, Spongilla, freshwater sponge, and Euspongia bath sponge. Phylum Coelenterata Cnidaria. They are aquatic, mostly marine, sessile or free-swimming, radially symmetrical animals. Figure 4.6. The name Cnidaria is derived from the cnidoblasts or cnidocytes, which contain the stinging capsules or nematocysts present on the tentacles and the body. Cnidoblasts are used for anchorage, defense, and for the capture of prey. Figure 4.7. Cnidarians exhibit tissue level of organization and are diploblastic. They have a central gastrovascular cavity with a single opening, mouth on hypostome. Digestion is extracellular and intracellular. Some of the cnidarians, for example corals, have a skeleton composed of calcium carbonate. Cnidarians exhibit two basic body forms called polyp and medusa. Figure 4.6. The former is a sessile and cylindrical form like Hydra, Edempsia, and so on, whereas the latter is umbrella-shaped and free-swimming like Aurelia or jellyfish. Those cnidarians which exist in both forms exhibit alternation of generations, metagenesis. That is, polyps produce medusae asexually, and medusae form the polyps sexually, for example, Lobelia. Examples: Zalia Portuguese man of war, Dempsia sea anemone, Inatula sea pen, Gorgonia sea fan, and Meandrina brain coral. Figure 4.6. Examples of Coelenterata indicating body forms: Aurelia medusa, B. Edempsia polyp. The figure contrasts the two structural forms found in cnidarians coelenterates. Medusa aurelia, the jellyfish form, umbrella-shaped, free-swimming, and adapted for locomotion in open water. Tentacles hang downward, equipped with cnidoblasts, specialized stinging cells for prey, capture, and defense. Polyp Edempsia, a cylindrical, sessile form attached to substrates like rocks or shells. Edempsia sea anemone displays a mutualistic association with hermit crabs, gaining mobility while offering protection to its host. This comparison illustrates the dimorphism of cnidarians, where both forms play distinct ecological roles and can alternate within a single life cycle, a phenomenon called metagenesis. Figure 4.7. Diagrammatic view of cnidoblast. This microscopic diagram depicts a single cnidoblast cell, a defining feature of cnidarians. The cnidoblast contains a capsule called a nematocyst, coiled inside like a harpoon. The trigger cnidocell responds to touch or chemical stimuli, causing the thread to explosively eject, delivering toxins to paralyze prey or deter predators. The cell's nucleus and cytoplasmic components are shown, emphasizing that cnidoblasts are not just defensive structures but also vital for feeding. This figure helps students visualize how such a tiny cellular adaptation has contributed to the ecological success of jellyfish, corals, and sea anemones in marine environments. Phylum Ctenophora. Ctenophores, commonly known as sea walnuts or cone jellies, are exclusively marine, radially symmetrical, diploblastic organisms with tissue level of organization. The body bears 8 external rows of ciliated cone plates, which help in locomotion, figure 4.8. Digestion is both extracellular and intracellular. Bioluminescence, property of a living organism to emit light, is well marked in ctenophores. Sexes are not separate. Reproduction takes place only by sexual means. Fertilization is external with indirect development. Examples: Pleurobrachia and Ctenoplana. Figure 4.8. Example of Ctenophora pleurobrachia. The figure presents Pleurobrachia, commonly known as the sea walnut, a representative of Phylum Ctenophora. The transparent, spherical body is adorned with 8 comb rows, bands of cilia that beat rhythmically to produce a shimmering effect, propelling the animal through water. The diagram shows 2 long retractile tentacles used for capturing planktonic prey. These animals are bioluminescent, producing soft light in the dark ocean through chemical reactions, an adaptation for communication and predator evasion. Ctenophores are important for showing the transition between simple cnidarians and more complex bilaterians, with features like a complete digestive tract and distinct tissues. Phylum Platyhelminthes. They have dorso-ventrally flattened body, hence are called flatworms, figure 4.9. These are mostly endoparasites found in animals, including human beings. Flatworms are bilaterally symmetrical, triploblastic, and acoelomate animals with organ level of organization. Hooks and suckers are present in the parasitic forms. Some of them absorb nutrients from the host directly through their body surface. Specialized cells called flame cells help in osmoregulation and excretion. Sexes are not separate. Fertilization is internal, and development is through many larval stages. Some members, like Planaria, possess high regeneration capacity. Examples: Tania tapeworm, Fasciola liver fluke. Figure 4.9. Examples of Platyhelminthes are tapeworm B, liver fluke. This figure illustrates parasitic flatworms, highlighting their dorsoventrally flattened bodies and adaptations for a parasitic lifestyle. Tapeworm Tania, a long ribbon-like worm consisting of repeated segments called proglottids, each containing male and female reproductive organs. The scolex head bears suckers and hooks for attachment to the intestinal wall of its host. Liver fluke fasciola, leaf-shaped and hermaphroditic. It lives in the bile ducts of cattle and sheep. It requires two intermediate hosts, a snail and an aquatic plant, for completing its life cycle. The figure emphasizes the loss of digestive and sensory systems in favor of reproductive and attachment structures, clear adaptations to endoparasitic existence. Phylum Aschelminthes. The body of the Aschelminthes is circular in cross-section. Hence, the name roundworms, figure 4.10, they may be free-living, aquatic and terrestrial, or parasitic in plants and animals. Roundworms have organ system level body organization. They are bilaterally symmetrical, triploblastic, and pseudocoelomate animals. Alimentary canal is complete with a well-developed muscular pharynx. An excretory tube removes body wastes from the body cavity through the excretory pore. Sexes are separate dioecious. As males and females are distinct. Often females are longer than males. Fertilization is internal, and development may be direct. The young ones resemble the adult or indirect. Examples: Ascaris roundworm, Wuchereria filaria worm, Ancylostoma hookworm. Figure 4.10 dames. Example of Aschelminthes, roundworm Ascaris. This figure shows the general structure of Ascaris lumbricoides, a parasitic roundworm belonging to the phylum Aschelminthes Nemathelminthes. Its body is elongated, cylindrical, and tapered at both ends, covered by a tough cuticle that protects it from digestive enzymes of the host. The sexes are separate dioecious, a key feature distinguishing this phylum. Males are smaller, with a curved posterior end, while females are larger and straight. The figure highlights a complete digestive system with distinct mouth and anus, marking an evolutionary step from incomplete to complete digestion. These worms inhabit the human intestine, causing ascariasis, and represent the advancement toward body organization with a pseudocoelom and distinct organ systems. Phylum Annelida. They may be aquatic, marine, and freshwater, or terrestrial, free-living, and sometimes parasitic. They exhibit organ system level of body organization and bilateral symmetry. They are triploblastic, metamerically segmented, and chelomate animals. Their body surface is distinctly marked out into segments or metameres, and hence the phylum name Annelida Latin. Annulus, little ring, figure 4.11. They possess longitudinal and circular muscles which help in locomotion. Aquatic annelids like Nereis possess lateral appendages, parapodia, which help in swimming. Closed circulatory system is present. Nephridia sing. Nephridium help in osmoregulation and excretion. Neural system consists of paired ganglia sing. Ganglion connected by lateral nerves to a double ventral nerve cord. Nereis, an aquatic form, is dioecious, but earthworms and leeches are monoecious. Reproduction is sexual. Examples: Nereis, Baratima earthworm, and Hirudinaria blood, sucking leech. Figure 4.11. Examples of Annelida. Nereis B. hirudinaria. This figure contrast to annelids, marine and parasitic, showing the versatility of segmented worm Phylum Annelida. Nereus, free-living marine worm, exhibiting metameric segmentation, the division of the body into ring-like segments both externally and internally. It possesses paired lateral appendages called parapodia for locomotion and respiration. Presence of closed circulatory system and well-developed nervous system indicates a higher level of organization. Hirudinaria leech, an ectoparasite found in fresh water, feeding on the blood of vertebrates. The figure highlights its suckers at both ends and the secretion of hirudin, an anticoagulant preventing blood clotting. Together, they illustrate the adaptive diversity within annelids, from active swimmers to parasitic forms, linked by their shared segmented body plan. Phylum Arthropoda. This is the largest phylum of Animalia, which includes insects. Over 2/3 of all named species on Earth are arthropods, figure 4.12. They have organ system level of organization. They are bilaterally symmetrical, triploblastic, segmented, and coelomate animals. Body of arthropods is covered by chitinous exoskeleton. The body consists of head, thorax, and abdomen, and have jointed appendages, arthros joint, poda appendages. Respiratory organs are gills, book lungs, or tracheal system. Circulatory system is of open type. Sensory organs like antennae, eyes compound and simple. Statocysts or balancing organs are present. Excretion takes place through Malpighian tubules. They are mostly dioecious. Fertilization is usually internal. They are mostly oviparous. Development may be direct or indirect. Examples: economically important insects, Apis honeybee, Bombyx silkworm, Laxifer lac, insect vectors, Anopheles, Culex, and Aedes mosquitoes, gregarious pest, Locusta locust, living fossil, Limulus king crab. Figure 4.12. Examples of arthropoda: a locust bee, butterfly C, scorpion D, prawn. This figure showcases representatives of the largest animal phylum, Arthropoda, distinguished by jointed appendages and a chitinous exoskeleton. Locust, a terrestrial insect with 3 distinct body regions: head, thorax, abdomen, and wings. Its ability to form swarms demonstrates social and behavioral adaptations. Butterfly displays striking metamorphosis from larva to adult, an adaptation for exploiting different habitats and food sources during life stages. Scorpion, an arachnid with segmented tail ending in a venomous sting, adapted for predation and defense. Prawn, crustacean with gills, for respiration and appendages specialized for swimming and feeding. Collectively, the figure represents the extraordinary adaptive radiation of arthropods in air, land, and water, making them the most successful group in the animal kingdom. Phylum Mollusca. This is the second largest animal phylum. Figure 4.13. Mollusks are terrestrial or aquatic, marine, or freshwater, having an organ system level of organization. They are bilaterally symmetrical, triploblastic, and coelomate animals. Body is covered by a calcareous shell and is unsegmented with a distinct head, muscular foot, and visceral hump. A soft and spongy layer of skin forms a mantle over the visceral hump. The space between the hump and the mantle is called the mantle cavity, in which feather-like gills are present. They have respiratory and excretory functions. The anterior head region has sensory tentacles. The mouth contains a file-like rasping organ for feeding, called radula. Figure 4.13. Examples of mollusca: Pila bee octopus. This figure highlights the diversity of soft-bodied animals under Phylum Mollusca. Pila apple snail is a coiled shell and a soft, unsegmented body divided into head, muscular foot, and visceral hump. The mantle secretes the shell and encloses the mantle cavity, which houses gills, cnidaria for respiration. Octopus. a shellless cephalopod showing extreme adaptation for an active predatory lifestyle. It has 8 muscular arms lined with suckers, a large brain, and well-developed eyes, signs of remarkable cephalization. This figure demonstrates how mollusks evolved from slow-moving shelled forms to intelligent, agile predators, representing a leap in neural and behavioral sophistication among invertebrates. They are usually dioecious and oviparous, with indirect development. Examples: Gila apple snail, Punctata pearl oyster, sepia cuttlefish, Loligo squid, octopus devilfish, Plesia sea hare, Dentalium tusk shell, and Chaetoplura chitin. Phylum Echinodermata. These animals have an endoskeleton of calcareous ossicles and, hence, the name Echinodermata spiny-bodied. Figure 4.14. All are marine with organ system level of organization. The adult echinoderms are radially symmetrical, but larvae are bilaterally symmetrical. They are triploblastic and coelomate animals. Digestive system is complete with mouth on the lower ventral side and anus on the upper dorsal side. The most distinctive feature of echinoderm is the presence of water vascular system, which helps in locomotion, capture, and transport of food and respiration. An excretory system is absent. Sexes are separate. Reproduction is sexual. Fertilization is usually external. Development is indirect with free-swimming larvae. Examples: Asterias starfish, Echinus sea urchin, Antedon sea lily, Hookamaria sea cucumber, and Ophiura brittle star. Figure 4.14. Examples of Echinodermata. Asterias B. Ophiura. This figure portrays typical members of phylum Echinodermata, which are exclusively marine and display radial symmetry in adults, bilateral in larvae. Asterias starfish has a 5-armed body radiating from a central disc. The figure emphasizes its water vascular system, used for locomotion, food capture, and respiration. Tiny tube feet protrude through the arms, enabling slow, powerful movement. Ophiura brittle star resembles a starfish but has slender, flexible arms distinctly set off from the central disc, adapted for quick, snake-like movements. The figure underscores echinoderm's unique features: endoskeleton of calcareous ossicles and regenerative ability that distinguish them as the most advanced invertebrates and close evolutionary relatives of chordates. Phylum Hemichordata. Hemichordata was earlier considered as a subphylum under Phylum Chordata, but now it is placed as a separate phylum under Non-Chordata. Hemichordates have a rudimentary structure in the collar region called stomochord, a structure similar to notochord. This phylum consists of a small group of worm-like marine animals with organ system level of organization. They are bilaterally symmetrical, triploblastic, and coelomate animals. The body is cylindrical and is composed of an anterior proboscis, collar, and a long trunk. Figure 4.15. Circulatory system is of open type. Respiration takes place through gills. Excretory organ is proboscis gland. Sexes are separate. Fertilization is external. Development is indirect. Examples: balanoglossus and saccoglossus. Figure 4.15. Balanoglossus. This figure depicts balanoglossus, commonly called the acorn worm, which belongs to the phylum Hemichordata, a group that bridges non-chordates and chordates. The body is divided into 3 distinct regions. Proboscis, a muscular, conical structure that helps in burrowing and food collection. Collar connects the proboscis and trunk, containing a short portion of the gut and the mouth. Trunk, the elongated posterior part housing the pharynx, gill slits, and reproductive organs. The figure emphasizes its gill slits and notochord-like structure restricted to the proboscis region, highlighting why hemichordates were once considered primitive chordates. It shows the evolutionary significance of balanoglossus as a connecting link between invertebrates and vertebrates, possessing both simple body organization and early chordate-like features. Chordata. Animals belonging to phylum Chordata are fundamentally characterized by the presence of a notochord, a dorsal hollow nerve cord, and paired pharyngeal gill slits, figure 4.16. These are bilaterally symmetrical, triploblastic coelomate with organ system level of organization. They possess a postanal tail and a closed circulatory system. Figure 4.16. Chordata characteristics. This diagram summarizes the three diagnostic features that define members of phylum Chordata. Notochord, a flexible, rod-like structure running along the body, providing skeletal support and serving as a primitive backbone in chordates. Dorsal hollow nerve cord, positioned above the notochord, it later develops into the brain and spinal cord in higher vertebrates. Pharyngeal gill slits, openings in the pharynx that function in filter feeding or respiration at some stage of development. The figure also indicates the postanal tail, which aids in movement and balance, and the endostyle or thyroid gland involved in filter feeding and metabolism. This schematic representation unifies all chordates, from simple Amphioxus to humans, under a shared structural blueprint that has been modified through evolution for different lifestyles. Table 4.1. Comparison of chordates and non-chordates. This table contrasts the key structural and functional differences between chordates and non-chordates in a point-by-point manner. First, notochord, present in chordates at least in the embryonic stage, but absent in non-chordates. Second, nerve cord, dorsal and hollow in chordate, ventral and solid in non-chordate. Third, circulatory system, closed in chordates, usually open in non-chordates. Fourth, heart position, ventral in chordates, dorsal or absent in non-chordates. And last, gill slits and postanal tail, present in chordates at some life stage, absent in non-chordates. This comparison table provides a comprehensive evolutionary perspective, showing how the emergence of the notochord and dorsal nerve cord marked the beginning of vertebrate lineage and greater physiological specialization. Phylum Chordata is divided into three subphyla: Urochordata or Tunicata, Cephalochordata, and Vertebrata. Subphyla Urochordata and Cephalochordata are often referred to as protochordates, figure 4.17, and are exclusively marine. In Urochordata, notochord is present only in larval tail, while in Cephalochordata it extends from head to tail region and is persistent throughout their life. Examples: Urochordata, Cydia, Salpa, Doliolum, Cephalochordata, Branchiostoma, Amphioxus, or Lancelet. The members of subphylum Vertebrata possess notochord during the embryonic period. The notochord is replaced by a cartilaginous or bony vertebral column in the adult. Thus, all vertebrates are chordates, but all chordates are not vertebrates. Besides the basic chordate characters, vertebrates have a ventral muscular heart with 2, 3, or 4 chambers, kidneys for excretion and osmoregulation, and paired appendages which may be fins or limbs. Figure 4.17. Cydia. The figure shows a Cydia sea squirt, a urochordate belonging to subphylum Tunicata. The adult form is sessile and sac-like, encased in a tunic made of tunicin, a cellulose-like substance. It has 2 siphons, incurrent and excurrent, for water flow through the body. Although the adult lacks a notochord and nerve cord, the larval stage possesses both, along with a postanal tail. This feature demonstrates the retrogressive metamorphosis, where the larva shows advanced chordate traits that are lost in the sedentary adult. The figure helps students understand the evolutionary reduction and specialization within chordates, emphasizing how developmental stages can reveal ancestral characteristics. Narration for the subphylum Vertebrata classification diagram. This diagram presents a clear overview of the classification of the subphylum Vertebrata, showing the evolutionary division based on the presence or absence of jaws and limbs. At the top, we have Vertebrata, which is divided into two major groups, Agnatha and Gnathostomata. 1. Agnatha. These are jawless vertebrates. The term Agnatha literally means without jaws. They represent the most primitive vertebrates, lacking a true jaw structure. The diagram shows that Agnatha contains only one class, Cyclostomata. Cyclostomes are eel-like, parasitic or free-living fishes such as lampreys and hagfishes. They possess a circular sucking mouth, a cartilaginous skeleton, and gill slits for respiration. 2. Gnathostomata. These vertebrates are characterized by the presence of jaws, an evolutionary advancement allowing more efficient feeding and predation. Gnathostomata is further divided into two major groups, Pisces and Tetrapoda, classified here as superclasses. Pisces fishes. These are aquatic vertebrates that bear fins. The diagram indicates two classes within Pisces. First, Chondrichthyes, cartilaginous fishes like sharks, rays, and skates, possessing a skeleton made of cartilage instead of bone. Second, Ostectides, bony fishes like Catla, Hippocampus, and others which have a bony endoskeleton, scales, and a swim bladder for buoyancy. Tetrapoda. This group includes vertebrates that bear limbs, enabling life on land or adaptations for terrestrial locomotion. Tetrapods are divided into four classes. First, amphibia, vertebrates like frogs and salamanders, which exhibit dual life, beginning in water and maturing on land, possessing moist skin for respiration. Second, reptilia, reptiles such as snakes, lizards, turtles, and crocodiles, adapted for life on land with scaly skin, internal fertilization, and amniotic eggs. Third, aves, birds characterized by feathers, beaks, hollow bones, and adaptations for flight. Fourth, mammals, warm-blooded vertebrates with hair, mammary glands for feeding young, and highly developed nervous and circulatory systems. This hierarchical diagram efficiently summarizes the evolutionary progression in vertebrates, from primitive jawless forms to advanced jawed, finned, and limbed vertebrates, highlighting how the acquisition of jaws and limbs shaped the diversity of life on Earth. Class Cyclostomata. All living members of the class Cyclostomata are ectoparasites on some fishes. They have an elongated body bearing -6 pairs of gill slits for respiration. Cyclostomes have a sucking and circular mouth without jaws. Figure 4.18. Their body is devoid of scales and paired fins. Cranium and vertebral column are cartilaginous. Circulation is of closed type. Cyclostomes are marine but migrate for spawning to fresh water. After spawning, within a few days, they die. Their larvae, after metamorphosis, return to the ocean. Examples: Petromyzon lamprey and myxine hagfish. Figure 4.18, a jawless vertebrate, Petromyzon. This figure represents Petromyzon lamprey, a jawless fish from the class Cyclostomata, the most primitive group of living vertebrates. The eel-like body is elongated, scaleless, and lacks paired fins. The circular suctorial mouth functions as an attachment organ, helping it cling to other fish and feed on their body fluids. The figure highlights the 7 pairs of gill openings, cartilaginous skeleton, and notochord persisting throughout life, features that signify its primitive vertebrate nature. Petromyzon bridges the evolutionary gap between invertebrates and jawed vertebrates, showcasing how early vertebrates evolved fundamental traits like a cranium and closed circulatory system before the evolution of jaws and paired limbs. Class Chondrichthyes. They are marine animals with streamlined body and have cartilaginous endoskeleton. Figure 4.19. Mouth is located ventrally. Notochord is persistent throughout life. Gill slits are separate and without operculum gill cover. Skin is tough, containing minute placoid scales. Teeth are modified placoid scales which are backwardly directed. Their jaws are very powerful. These animals are predaceous. Due to the absence of air bladder, they have to swim constantly to avoid sinking. Figure 4.19. Examples of cartilaginous fishes. Ascoliodon B. pristis. This figure represents two members of the class Chondrichthyes, also known as cartilaginous fishes, characterized by a skeleton made entirely of cartilage. Ascoliodon dogfish exhibits a streamlined, spindle-shaped body adapted for active swimming. The figure highlights paired fins, a heterocercal, unequal-lobed tail for propulsion, and 5 pairs of gill slits without an operculum. Its mouth is ventrally placed and skin is covered with placoid scales which reduce friction and offer protection. B. pristis sawfish, noted for its elongated snout with tooth-like projections resembling a saw, used for defense and stirring up prey. Both examples demonstrate the adaptive evolution of primitive vertebrates for aquatic predation, possessing cartilaginous endoskeletons, powerful jaws, and well-developed sensory organs, but lacking a swim bladder. Hence, they must swim continuously to avoid sinking. Heart is two-chambered, one auricle, and one ventricle. Some of them have electric organs, for example, torpedo, and some possess poison sting, for example, trigon. They are cold-blooded poikilothermous animals. That is, they lack the capacity to regulate their body temperature. Sexes are separate. In males, pelvic fins bear claspers. They have internal fertilization, and many of them are viviparous. Examples: Goliodon dogfish, pristis sawfish, Parcherodon, great white shark, trigon stingray. Class Chondrichthyes. It includes both marine and freshwater fishes with bony endoskeleton. Their body is streamlined. Mouth is mostly terminal, figure 4.20. They have 4 pairs of gills, which are covered by an operculum on each side. Skin is covered with cycloid ctenoid scales. Air bladder is present, which regulates buoyancy. Heart is two-chambered, one auricle and one ventricle. They are cold-blooded animals. Sexes are separate. Fertilization is usually external. They are mostly oviparous and development is direct. Examples: marine, Exochetus flying fish, hippocampus, seahorse, freshwater, labeo rohu, catla, Clarius mager, aquarium. Betta fighting fish, Pterophyllum angelfish. Figure 4.20. Examples of bony fishes, a hippocampus bicatla. This figure showcases members of the class Osteichthyes, or bony fishes, which are characterized by a bony endoskeleton, cycloid or ctenoid scales, and an operculum covering gill slits. The hippocampus seahorse possesses an upright posture and curved tail for clinging to vegetation. Interestingly, in this species, males carry the fertilized eggs in a brood pouch until hatching, a rare reproductive adaptation. B. catla, freshwater fish commonly found in Indian rivers. Known for its deep body and large head. It has a terminal mouth, operculum-covered gills, and a swim bladder, helping it maintain buoyancy. The figure emphasizes how bony fishes display enormous diversity and advanced features such as air bladder regulation, external fertilization, and streamlined bodies suited for rapid movement and stability in various aquatic environments. Class Amphibia. As the name indicates, gender, amphi, dual, bios, life, amphibians can live in aquatic as well as terrestrial habitats. Figure 4.21. Most of them have 2 pairs of limbs. Body is divisible into head and trunk. Tail may be present in some. The amphibian's skin is moist without scales. The eyes have eyelids. A tympanum represents the ear. Alimentary canal. Urinary and reproductive tracts open into a common chamber called cloaca, which opens to the exterior. Respiration is by gills, lungs, and through skin. The heart is 3-chambered 2 auricles and 1 ventricle. These are cold-blooded animals. Sexes are separate. Fertilization is external. They are oviparous and development is indirect. Examples: bufo toad, rana frog, hyla tree frog, salamandra salamander, ichthyophis, limbless amphibia. Figure 4.21. Examples of amphibia. A salamandra, B. rana. This figure depicts members of class Amphibia, representing the first vertebrates to live both in water and on land. A salamandra, salamander, a lizard-like amphibian showing tail retention in adults and smooth, moist skin essential for cutaneous respiration. B. rana frog, common amphibian adapted for dual life. Its webbed feet aid in swimming, while long hind limbs support leaping on land. The figure demonstrates how amphibians bridge aquatic and terrestrial habitats, with features such as three-chambered hearts, glandular skin for respiration, and external fertilization. Their life cycle includes metamorphosis, transitioning from gill-breathing larvae to air-breathing adults, marking a major evolutionary leap toward terrestrial life. Class Reptilia. The class name refers to their creeping or crawling mode of locomotion, Latin repere or reptum, creep or crawl. They are mostly terrestrial animals and their body is covered by dry and cornified skin, epidermal scales or scutes, figure 4.22. They do not have external ear openings. Tympanum represents ear. Limbs, when present, are two pairs. Heart is usually three-chambered, but four-chambered in crocodiles. Reptiles are poikilotherms. Snakes and lizards shed their scales as skin cast. Sexes are separate. Fertilization is internal. They are oviparous and development is direct. Examples: Pelone turtle, Testudo tortoise, chameleon tree lizard, Pelotes garden lizard, Crocodilus crocodile, alligator alligator, Hemidactylus wall lizard, poisonous snakes, Naja cobra, Bangurus crite, Vipera viper. Figure 4.22. Reptiles, chameleon bee, Crocodilus C. chelone D. Naja. This figure exhibits diverse members of class Reptilia, which evolved complete adaptation to land through scaly, water-resistant skin and internal fertilization. A chameleon, arboreal reptile with a prehensile tail, independently movable eyes, and color-changing skin. Adaptations for camouflage and hunting. Crocodilus, a semi-aquatic reptile with a strong tail and bony scutes. It exhibits four-chambered heart and parental care, both advanced reptilian traits. C. Pelone turtle. Possesses a bony shell carapace and plastron enclosing the body, offering protection. D. Naja cobra. A venomous snake capable of spreading its hood when threatened, used for both defense and intimidation. Together, the figure shows the evolutionary diversification of reptiles, with physiological adaptations like lungs for respiration, amniotic eggs, and ectothermic regulation, allowing them to thrive in terrestrial habitats. Class Aves. The characteristic features of Aves birds are the presence of feathers, and most of them can fly except flightless birds, for example, ostrich. They possess beak, figure 4.23. The forelimbs are modified into wings. The hind limbs generally have scales and are modified for walking, swimming, or clasping the tree branches. Skin is dry, without glands except the oil gland at the base of the tail. Endoskeleton is fully ossified bony, and the long bones are hollow with air cavities pneumatic. The digestive tract of birds has additional chambers, trap and gizzard. Heart is completely four-chambered. They are warm-blooded homoeothermous animals. That is, they are able to maintain a constant body temperature. Respiration is by lungs. Air sacs connected to lungs supplement respiration. Sexes are separate. Fertilization is internal. They are oviparous, and development is direct. Examples: Corvus crow, Palumba pigeon, Psittacula parrot, Druthio ostrich, Avo peacock, Captain Heights penguin, Neophron vulture. Figure 4.23. Some birds, a Neophron bee, Druthio C. Psittacula D. pavo. This figure presents representatives of class aves, highlighting their structural adaptations for aerial life. A neophron vulture, a scavenger bird with broad wings for soaring flight and a strong beak for tearing flesh. B. Struthio ostrich, the largest living bird, flightless but specialized for running, with strong legs and reduced wings. C. Tacacula parrot, possesses a curved beak and zygodactyl feet for climbing and grasping, known for intelligence and vocal mimicry. D. Pavo peacock, national bird of India, noted for its brilliant plumage and ornamental tail feathers used in courtship displays. The figure collectively illustrates avian features, feathers, light bones, wings, and warm-blooded metabolism, which evolved for efficient flight and thermoregulation, marking birds as highly specialized vertebrates. Class Mammalia. They are found in a variety of habitats: polar ice caps, deserts, mountains, forests, grasslands, and dark caves. Some of them have adapted to fly or live in water. The most unique mammalian characteristic is the presence of milk-producing glands, mammary glands, by which the young ones are nourished. They have 2 pairs of limbs, adapted for walking, running, climbing, burrowing, swimming, or flying. The skin of mammals is unique in possessing hair. External ears or pinnae are present. Different types of teeth are present in the jaw. Heart is 4-chambered. They are homoeothermous. Respiration is by lungs. Sexes are separate and fertilization is internal. They are viviparous with few exceptions and development is direct. Examples: Oviparous Ornithorhynchus platypus, Viviparous Macropus kangaroo, Paropus flying fox, Camelus camel, Macaca monkey, Rattus rat, Canis dog, Phallus cat, Elephas elephant, Equus horse, Delphinus common dolphin, Balanoptera blue whale, Panthera tigris tiger, Panthera Leo lion. Figure 4.24. Some mammals: A. Ornithorhynchus, B. Macropus, C. Pteropus, D. Balanoptera. This figure illustrates the diversity and adaptive specialization within class Mammalia, the most advanced class of vertebrates. The Ornithorhynchus platypus, monotreme or egg-laying mammal. It exhibits primitive mammalian features like egg-laying and cloaca, alongside advanced traits like mammary glands for lactation. The duck-like bill is electroreceptive, helping it locate prey underwater. B. Macropus kangaroo, a marsupial that carries young in a ventral pouch. Adapted for terrestrial life, it has powerful hind limbs for hopping and long tail for balance. This figure demonstrates reproductive specialization and locomotor adaptation. C. Paropus flying foxbat, placental mammal capable of powered flight. The wings are modified forelimbs, while the body is covered with fur for insulation. The figure highlights the ecological role of bats in pollination and seed dispersal, showing behavioral and anatomical adaptations. D. Balanoptera blue whale represents aquatic mammals with adaptations for marine life, including streamlined body, blubber for insulation, and modified forelimbs as flippers. The whale shows evolutionarily advanced placental reproduction and complex social behavior, bridging terrestrial and aquatic adaptations in mammals. This figure collectively emphasizes mammalian diversity, highlighting reproductive strategies, locomotion, feeding adaptations, and ecological niches, marking mammals as highly evolved vertebrates with complex physiology and behavior. The salient distinguishing features of all phyla under Animal Kingdom is comprehensively given in the Table 4.2. Table 4.2. Salient features of different phyla in the Animal Kingdom. Let's explore the major animal phyla and their distinguishing characteristics, as summarized in this table. We start with the simplest animals and move toward the more complex forms, highlighting their organization, symmetry, body cavity, and unique adaptations. Porifera sponges. These are the most primitive multicellular animals. They are asymmetrical or sometimes show radial symmetry. Their bodies are cellular level of organization without true tissues or organs. Porifera are acoelomate, meaning they have no body cavity. They live mostly in marine environments and are sessile filter feeders, drawing water through pores to obtain food. Coelenterata, Cnidarians. These animals exhibit radial symmetry and have a tissue-level organization with two germ layers, ectoderm and endoderm. They are diploblastic and acoelomate, possessing a gastrovascular cavity for digestion. Coelenterates include hydra, jellyfish, and corals, and they have cnidoblasts, specialized stinging cells used for defense and prey capture. Platyhelminthes, flatworms. Platyhelminthes are bilaterally symmetrical and triploblastic, showing organ-level organization. They are acoelomate, lacking a body cavity between the gut and body wall. Examples include planaria, tapeworms, and liver flukes. They exhibit cephalization, primitive digestive system, and adaptations for both free-living and parasitic lifestyles. Aschelminthes, or nematode roundworms. Roundworms are bilaterally symmetrical and triploblastic, with pseudocoelom, body cavity not fully lined with mesoderm. Their bodies are elongated and cylindrical with a complete digestive system. Many, like askers, are parasitic, while others are free-living. They have separate sexes and exhibit simple excretory and nervous systems. Annelida, segmented worms. Annelids show bilateral symmetry, triploblastic organization, and are chelomates, meaning they possess a true body cavity lined with mesoderm. Their bodies are metamerically segmented, each segment containing repeated organs and structures. Examples include earthworms, leeches, and nereis. Annelids have a closed circulatory system, a well-developed nervous system, and excretory structures called nephridia. Arthropoda insects, crustaceans, and arachnids. This largest animal phylum exhibits bilateral symmetry, triploblastic organization, and coelomate body cavity. Arthropods have a chitinous exoskeleton, jointed appendages, and segmented bodies. Examples include insects, scorpions, crabs, and prawns. They have a well-developed nervous system, compound eyes, and exhibit remarkable adaptations for terrestrial, aerial, and aquatic habitats. Mollusca snails, octopus, clams. Mollusks are bilaterally symmetrical, triploblastic, and coelomate. They have a soft, unsegmented body divided into head, foot, and visceral mass, often protected by a calcareous shell. Examples include Gila, octopus, and clams. They possess a mantle, complete digestive system, open or closed circulatory system, and well-developed sensory organs in some species. Echinodermata, starfish. Sea urchin. Echinoderms are radially symmetrical as adults, but their larvae are bilaterally symmetrical, reflecting evolutionary transition. They are triploblastic and coelomate, with a calcareous endoskeleton and a unique water vascular system used for locomotion and feeding. Examples include Asterias and Ophiura. They have a complete digestive system, lack excretory organs, and show remarkable regeneration ability. Hemichordata balanoglossus. Hemichordates are bilaterally symmetrical, triploblastic, and coelomate. They show a dorsal nerve cord and gill slits. The notochord is limited to the proboscis region. Balanoglossus is a burrowing marine worm demonstrating a combination of invertebrate and chordate features, representing an evolutionary link. Chordata fishes, amphibia, reptiles, birds, mammals. Chordates are bilaterally symmetrical, triploblastic, and coelomate, characterized by a notochord, dorsal hollow nerve cord, pharyngeal gill slits, postanal tail, and endostyle or thyroid gland. Subphyla include Urochordata, Cephalochordata, and Vertebrata. Vertebrates exhibit complex organ systems, advanced nervous and circulatory systems, and adaptations to diverse habitats, ranging from aquatic to terrestrial and aerial environments. This table gives us a clear overview of the evolutionary progression of animals, showing how structural complexity, body cavity development, symmetry, and organ systems evolved. from simple sponges to highly specialized vertebrates. Summary. The basic fundamental features such as level of organization, symmetry, cell organization, coelom, segmentation, notochord, and so on, have enabled us to broadly classify the animal kingdom. Besides the fundamental features, there are many other distinctive characters which are specific for each phyla or class. Porifera includes multicellular animals which exhibit cellular level of organization and have characteristic flagellated choanocytes. The Coelenterates have tentacles and bear cnidoblasts. They are mostly aquatic, sessile, or free-floating. The ctenophores are marine animals with comb plates. The platyhelminthes have flat body and exhibit bilateral symmetry. Parasitic forms show distinct suckers and hooks. Aschelminthes are pseudocoelomates and include parasitic as well as non-parasitic roundworms. Annelids are metamerically segmented animals with a true coelom. The arthropods are the most abundant group of animals characterized by the presence of jointed appendages. The mollusks have a soft body surrounded by an external calcareous shell. The body is covered with external skeleton made chitin. The echinoderms possess a spiny skin. Their most distinctive feature is the presence of water vascular system. The hemichordates are a small group of worm-like marine animals. They have a cylindrical body with proboscis, collar, and trunk. Phylum Chordata includes animals which possess a notochord either throughout or during early embryonic life. Other common features observed in the chordates are the dorsal, hollow nerve cord, and paired pharyngeal gill slits. Some of the vertebrates do not possess jaws agnatha, whereas most of them possess jaws Gnatha, stomata. Agnatha is represented by the class Cyclostomata. They are the most primitive chordates and are ectoparasites on fishes. Gnathostomata has two superclasses, Pisces and Tetrapoda. Classes Chondrichthyes and Osteichthyes bear fins for locomotion and are grouped under Pisces. The Chondrichthyes are fishes with cartilaginous endoskeleton and are marine. Classes Amphibia, Reptilia, Aves, and Mammalia have two pairs of limbs and are thus grouped under Tetrapoda. The amphibians have adapted to live both on land and water. Reptiles are characterized by the presence of dry and cornified skin. Limbs are absent in snakes. Fishes, amphibians, and reptiles are poikilothermous cold-blooded. Aves are warm-blooded animals with feathers on their bodies and forelimbs modified into wings for flying. Hind limbs are adapted for walking, swimming, perching, or clasping. The unique features of mammals are the presence of mammary glands and hairs on the skin. They commonly exhibit viviparity. You've just finished listening to Chapter 4, Animal Kingdom from NCERT Class 11 Biology here on BioDecode. I hope this audio helped you clearly understand the key concepts and made exploring the vast diversity of animals more engaging. From simple body plans to complex organ systems, we've examined how animals are classified, the biological criteria used, and the evolutionary adaptations that define each group. In the next episode, we'll go through the exercise solutions and important questions from this chapter, explaining each answer step by step to help you prepare confidently for your exams. So make sure to follow BioDecode and stay tuned for the next part of this series. Keep learning, keep exploring, and I'll meet you in the next audiobook, Animal Kingdom, NCERT, Solutions and Key Questions. And yes, students, don't forget to like, share, and subscribe to our channel, BioDecode. See you in the next audio lecture.